speaker, Dr. Graciela Diaz de Delgado from Universidad de los Andes in Venezuela, and she's going to make the presentation Synchrotron powder diffraction for the characterization of pharmaceuticals. So we're very glad to have you with us. Dr. Graciela, the floor is yours.
Thank you very much, Carolina. I'm going to share my screen, start sharing my screen. I hope you can see it.
Presentation is preparing. Now we can see it, but in presentation mode.
Okay.
Everything is opening. And when it's ready, now we can see it in presentation mode. Thank you very much, Graciela. You can go ahead.
Okay, but I don't see it. I don't know why. We just, let me see.
Sometimes with F8,
Okay,
it can
let
be
me
shared
see.
into
Yeah,
your
probably.
screen as well.
Okay, yeah, I went to the other screen. Okay, I think now I can see it also.
Fantastic.
Okay, well,
go ahead, please.
Yeah, thank you very much, Carolina. It's a pleasure for me to participate this time as an instructor in this very prestigious school. I will try to summarize a little bit of what we need to keep in mind for powder diffraction, and then give just a few examples of the work that we did as part of a LAMP award as a fast team some years ago. But first, I would like to acknowledge my collaborators, my closest collaborators, Professor Miguel Delgado and Professor Analio Dugarte from Universidad de los Andes here in Venezuela, Dr. Robert Toro and Professor José Antonio Henao from Universidad Industrial de Santander in Bucaramanga, Colombia, Andy Fish and Catherine DeJoy from ESRF in France, and Jakob van de Strik from Avantgarde in Germany. I think we have been working as a very good team in recent years, and we are all very glad to be able to share some of the work that we have been doing recently. Okay, and also because it's the first time that I make a presentation of this type, I will just like to show you very briefly where I am located. I am in Mérida, far from the capital, actually closer to the border with Colombia. Mérida is one of the Andes states, and the university has 3 campuses, and the main campus is in Mérida. It has 2 other campuses in the other Andean states of Venezuela. The city is in a raised valley at about 1,600 meters above sea level, and we have usually we have a little bit snow in the mountains, and this is a view of the city. The university started as a seminary in 1785 and later became a university in 1810, soon after the Declaration of Independence, and then it became Universidad de los Andes. Our Faculty of Sciences was founded in 1967. This is a view of the building and some of the mountains, and our lab starts here, from here. So we have I think we have a good laboratory space. Our laboratory was founded in 1969, and since then we have been trying to carry out the best possible work with the resources available to us. The university is not only the city is not only a university city, but it's also a very touristic place. It's very well visited by Venezuelans and people from abroad. And one of our major attractions is the highest and the second longest cable car system in the world. And this is some of the typical views that we enjoy around here, mountains with a little bit of snow, and the very emblematic Speletia plant that it grows above 3,000 meters in this region. Okay, so I would like to start by pointing out 4 milestones in X-ray diffraction. Of course, we can think about many different contributions, but we think that there are basically 4 milestones that need to be remembered. If First of all, of course, the discovery of X-rays in 1895 by Wilhelm Conrad Röntgen, who obtained the Nobel Prize in Physics for his discovery. The second one, the discovery of X-ray diffraction by crystals in about 1912. This was an experiment carried out by Walter Friedrich and Paul Nippen, but the experiment was suggested by Max von Laue. Max von Laue received the Nobel Prize in physics, but he shared it with Friedrich and Nippen because of they did a very nice experimental work. Certainly, a major contribution was the diffraction by single crystals made by William Henry and William Lawrence Bragg, father and son, that also made them recipients of the Nobel Prize in Physics this time in 1915. And pretty much at the same time, around 1916, in Europe, Peter Debye and Paul Scherrer, and in the US, Albert Hall, developed the methods for powder diffraction using X-rays. This is a very, very important contribution because of the nature or the application that powder diffraction has had over the years in the characterization of materials. And we are going to emphasize in this talk some of the most important aspects of this technique. Let's first remember that what we consider to be crystalline materials. For a crystalline material, we need a regular arrangement of atoms and molecules in some kind of repetition pattern in 3 dimensions. We call conventional crystalline materials to those compounds that have a regular and periodic arrangement of those atoms and molecules. When we have an irregular and aperiodic arrangement of those motifs, we have the so-called amorphous materials. There is a very important group of materials that are regular but aperiodic, among which we find the quasi-crystals. And Dan Schenkman received the Nobel Prize in Chemistry in 2011 because of the description of these materials. But we are not going to talk about this type of compound, which are very interesting, but we are not going to deal with them in this talk. So basically what we have, or what we like to have, is some kind of a structural motif, which could be atoms or molecules, or a complex, an organometallic complex that is repeated according to a particular pattern that is called the crystal lattice, and to obtain a regular arrangement of those motifs, atoms or molecules in 3 dimensions. That arrangement can be described in terms of a small parallelepiped, which is called the unit cell, that when we reproduce that small unit with a unit that is characterized by the smallest volume possible and the highest symmetry possible, when we repeat that unit in 3 dimensions, we reproduce the complete crystal structure. So traditionally, we consider conventional crystalline materials those materials that are that have a constituent atoms or ions or molecules in a regular and periodical 3-dimensional arrangement in the space. In that arrangement, we define what is called a unit cell that is described in terms of 3 vectors and the angles between them. And when we repeat this basic unit, we reproduce the whole crystal structure. One way to understand the interaction of X-rays and actually the radiation with an arrangement of this kind is through the interpretation that William Lawrence Bragg, gave to the diffraction phenomena. Of course, there is a very formal and very more elaborate ways to present this, but I think for the purposes of this talk and for a general understanding of the subject, the Bragg interpretation is very, very convenient. So what William Lawrence Bragg proposed, was that if we have a set of X-rays, of X-ray beams that are in interaction with the planes, crystalline planes that we can define in that unit cell, they will be diffracted, or in a similar way as a reflection of light by a mirror, they will be reflected with the same angle of incidence. Then we can register that reflected beam. But at the same time, there are many other rays that are interacting with the same with that particular set of planes. So some of them will be in phase, partially in phase, or completely out of phase. For those emerging beams that have a path difference, which is an integer number of the integer multiple of the wavelength, then we have met the diffraction condition. Those rays will be in phase, and we will produce a reflection coming from that particular set of planes that are separated by a distance d. We can see that also in a simplified way here. We have a set of planes, of crystalline planes, that are interacting with an X-ray beam that it's made to hit those particular planes with at an angle of theta, and when the diffracted beam emerges also at that theta angle, we have a reflection. Those planes are identified with their Miller indices, HKL, which are described in terms of the inverse of the intercept of that particular plane with different important crystallographic direction in the structure. Here we have an animation of what happens, and we can see that when to obtain a reflection, the rays have to be in phase so that Bragg's law is obeyed. Depending on the way we have the sample, we may have single crystals, or we have an aggregate of polycrystalline material. In the case of single crystal diffraction, we have just one fragment, one single crystal that we use, that we locate in the X-ray beam, and a series of reflections from the different crystalline planes in that structure are produced and registered in nowadays in 2D detectors. So we can have hundreds to thousands of reflections produced by the interaction of that X-ray beam. Actually can be also neutrons and electrons with a crystal that is being used in the experiment. In powder diffraction, we have an aggregate of a large number of very, very small single crystals. And also we use an X-ray beam that interacts with a specimen now composed of many very small single crystals, and what is produced is a one-dimensional, or what is registered at the end is a one-dimensional um, result, by a pattern, resulting pattern, which contains a few maxima, and some of those maxima may be superimposed with reflections from different sets of plane. So that's one of the main characteristics of the powder diffraction pattern. One way to understand the diffraction phenomena is the interpretation that was proposed by Paul Peter Ewald, based on the construction of the reciprocal lattice and the Ewald sphere, and the idea that Bragg's law is obeyed, is holds under certain geometric conditions. For example, if we have the cartoon that we had in the previous slides, where we have a set of crystalline planes, and I apologize because these names are in Spanish, but we have a set of crystalline planes identified by their indices HKL, and we have an incident X-ray beam, we have a reflected beam at the same theta angle. If we consider that the center of that sample is in a circle that is defined by the inverse of the wavelength that is used in the experiment, then it can be shown mathematically here. I'm not going to describe it in detail. It can be shown that every time that emerging beam touches the circle, which actually by rotation it can be made a sphere, touches a sphere constructed in this way, called the Ewald sphere, then a Bragg law is obeyed, and we obtain a diffracted beam that we can register with one available detector. So the idea is to have a lattice constructed based on the inverse of the spacing between the different set of planes, the so-called reciprocal lattice, and that lattice in some way is rotated to make the different planes that describe that lattice touch the sphere of reflection. And at that point, Bragg's law is obeyed, and we can see a reflection. So with the positions of those maxima, which are the theta HKL, we can calculate the spacing between the planes that are producing that particular reflection. And we can, from there, we can calculate the unit cell parameters of our crystalline material. This is another small movie representing how the different diffracted X-ray beams are obtained when we bring the reciprocal lattice to touch the surface of the sphere constructed in the way that Ewald proposed. So we have a characteristic pattern like this one that we register. This is actually a single crystal pattern, but something similar happens in powder diffraction. Basically, what we need is to have an X-ray source, some goniometer, some device that will bring the different planes, the different reciprocal lattice planes, to touch the sphere of reflection, somehow a detector, and all these components controlled by a system that can bring different crystalline planes to follow the diffraction condition that is governed by Bragg's law. So that's the basic thing with the diffractometer. In that sense, the direction of the beam is important because it will give us the size and shape of the unit cell, and the intensity of the beam is important because it is related to the nature of the atoms and molecules that are producing that reflection, and the spatial position of the different of the different atoms in those planes. So at the end, that would be the way to obtain the crystalline structure of that material. We have different types of X-ray sources, the conventional, all X-ray tubes, more recent sources as microfocus and different types of arrangements. We have synchrotron sources. This is a view of Sirius synchrotron in Brazil and a view of ESRF in Grenoble. And we have also some other emerging technologies like F-spheres that promise and have already demonstrated that are very useful in very particular and important cases. For the way to bring all the different planes to follow the diffraction condition, we need somehow a way to bring those planes, and that's why there are different geometries of goniometers have been devised. These are two goniometers from different commercial vendors, and but they use the Bragg-Brentano geometry, which is the most common one in a laboratory setting. For the synchrotron, this is a view of the Hodge at I-beam ID22 beam line at Grenoble, and here we have a view of the diffractometer, the detector, some device for low temperature analysis, a robot to put the samples, etc. Here we have a more recent view of this goniometer with a newer detector, and we have here an amplified view of the sample in a capillary mounted for synchrotron data collection. As for the detectors, we have point detectors, linear detectors that are useful, but of course they give a limited portion of the reciprocal space, but are nevertheless useful. And we also have area detectors where we can collect in a more rapid way several reflections at the same time. Basically, the information contained in a powder diffraction pattern, we can visualize it here. First of all, we have the position of the maxima, which have to be recorded in the best possible way and are related to the unit cell, the size and the shape of the unit cell that describe that the particular arrangement, the particular crystalline arrangement of one or more crystalline components in the sample. We also have the relative intensities of those reflections that is related to the type and the position of the atoms in the crystalline arrangement. We have the shape of the maxima that is related to the microstructure, for example, the different crystalline domains, changes in the shape due to tensions, defects in the structure, etc. And we have the background that includes a lot of information which is nevertheless important because it could indicate the presence of amorphous phases, the presence of diffuse scattering, another type of a low crystallinity components, for example. One of the major problems that we can encounter when we use powder diffraction, particularly in the Bragg-Brentano geometry, is it's the presence of several sources of errors. And for structural determination from powder diffraction, the most important one is the preferred orientation. And this is something that needs to be taken well care of, for example, in the preparation of the sample, a correct preparation of the sample, and there is also ways to try to assess that through some different algorithms. Just to show a little bit of what it looks like, if we have no preferential orientation in a sample, which is the ideal situation, all planes are detected equally, and we have a true registry of a good registry of the intensities of each reflection. However, if we have a preferred orientation, if the crystallites tend to orient in a particular way, as it's shown here, then we could have an effect on the intensities of certain reflections that come from different planes. So that's something that is very important and is very critical in Bragg-Brentano geometry. Here, just to summarize some of the most important applications of X-ray powder diffraction, we have the identification of different crystalline phases in a sample, quantification of crystalline phases, and the structural analysis of different materials, which has become more and more easier to carry out because of different advances in instrumentation and in the programs that deal with this type of information. So the most common application of powder diffraction is the identification of crystalline components, of components in a polycrystalline aggregate. And in this case, we have a very a nice review article in powder diffraction. Here is the link on the contribution of Albert Hall in the US. The most common application, I mean, it's very important to identify correctly the components of a polycrystalline aggregates, and this is also a representation of that particular case. For example, we have for silicon dioxide several phases. One is amorphous, and we have other phases with different crystal structures, and the power of the powder diffraction methods is that it allows us to identify each crystalline phase because each crystalline phase has a characteristic pattern. And also in a mixture of crystalline phases, each phase produces a particular pattern that is independent on the other phases. So it allows not only identification, qualitative identification, but also quantification of the presence of those different components. The identification process can be summarized here. Each powder diffraction pattern is characteristic of a material, so it is we can say that it's a fingerprint, and we can identify several basic steps. We have a search of the diffraction pattern, usually in a database, a superposition or in-house or commercial databases, a superposition of that particular experimental pattern with a reference pattern that is found in a database, the identification, deciding if the superposition of those patterns is appropriate and if they correspond to a phase that is already published, and repeat the process for any other phases that are present. And at the end, if possible, quantify the presence of each of the crystalline phases. For that, we have very good databases, in particular for powder diffraction. The PDF5 Plus database is the database to use for the identification of materials. The Cambridge Structural Database contains mostly single crystal structural determinations, but the powder diffraction patterns can be simulated from single crystal data, and they can also be used in conjunction with the PDF5 Plus database to identify materials. There is also the COD database, which contains a more limited number of phases, and in some cases, the quality of the patterns is not uniform across the database, but it may be useful for some applications and for some researchers. So the powder diffraction file is I would say this the standard to use to identify powder materials in a polycrystalline sample, and it started as a simple book, as a simple registry by hand at the beginning, and became after 1978 as the ICDD powder diffraction file. As I mentioned before, the COD database is an open database that has very good patterns for some materials. And here is a summary. I'm not going to go into those details, but it's a summary of the power of the powder diffraction file for the characterization of materials. And the Cambridge Structural Database, which even though it is basically a single crystal powder diffraction, a single crystal file or database, can be used to obtain, and here I have highlighted the programs and the algorithms that are used in the that can be used in powder diffraction. So the structural analysis using powder diffraction data may involve, for example, the identification of known phases. If we have, after registering the powder diffraction pattern, we can identify the crystalline phases using the powder diffraction file or the Cambridge Structural Database, or the ICSD, also the Inorganic Chemistry Database. And if we detect an isostructural phase, use that phase, use the so-called CIF file in a structural refinement of that phase using several programs. Some of them are public domain, and some of them are have a cost, but nevertheless, they are all important. If we have unknown phases, then we have to go through an indexing procedure using we like DICPOL, the program DICPOL, to determine the unit cell parameters. Then we need to do having a step-by-step profile of the pattern, do a pattern fitting and other decomposition procedures. We can use the several methodologies there that are properly referenced to. We need to somehow study the systematic presence of reflections to assign the space group. And we can carry out a structural determination process using different programs that use different algorithms, for example, DASH, EXPO, JANA, and other programs that are available. The structural refinement usually by the Riedel method and using the programs that we mentioned before. The structural refinement using Riedel, we have summarized here. I'm not going to go into the detail here because the lecture will be recorded. But this is basically what we have to do to obtain the unit cell, the nature and the position of the atoms that will describe the crystal structure, identify if there is more than one component, identify the quantitative ratio of all those components, and if necessary, if appropriate, study the microstructure, which is determine the crystallite size and any microstructural defects that might have the sample may have, or the specimen may have. And here we have tried to combine or to present what would be the workflow for a powder sample to go from sample preparation, a sample mounting, either in a flat sample holder or in capillaries, as it is used in mostly in the synchrotron, in high resolution synchrotron radiation. Collect the diffraction pattern. I try to identify the phases using the available databases. If there are no entries on those databases, then we need to go through an indexing, profile fitting, try to use some previously known information on the molecule, or use geometry optimization programs to try to define structural model to start with, go through a structural determination program, the process, using different methods that are shown here, structural refinement, and hopefully determine the crystal structure from powder diffraction data. Visualization, there are several programs, some free, some are with a license for visualization. Validation and DFT calculation is also very important, and different software for analysis of the material like Platon, Mogul, that allow, for example, the assessment of whether the distances and angles that have been obtained in that structural determination procedure are valid or not, if they make sense or not. And of course, different types of studies of intermolecular interactions. These are some key references, some very important books that we need to keep in mind, but the key reference is the volume H of the international tables for X-ray for crystallography. And here is a summary and outline of all the different parts. So this is a very important and very key reference for powder diffraction. There is also a very nice review in Nature Review Methods Primers by a very experienced group of powder diffractionists. Okay, so I'm just going to use a few minutes to talk about some of the work that we carried out at ESRF at Beamline ID 22 with Andy Fish and Catherine DeJoy in October to December of 22, and in 2023 we received a grant, and Alio and I received a grant through the LAMP project to carry out a study, and we took to ESRF some of the problems that we had encountered in our daily work that we could not solve satisfactorily with laboratory powder diffraction data. Just to remind what's the interest of powder diffraction in the pharmaceutical industry, at different points within in the research and development or in the production stages of a particular drug, it is important to characterize it by different methods, but X-ray diffraction is very, very important in the characterization of pharmaceutical compounds. For example, from the R&D viewpoint, at the laboratory stages, we need to characterize properly the different active pharmaceutical ingredients, determine if they present polymorphism, if they are obtained as salts or hydrates or any other kind of solvate, and any other structural feature that may be important for the characterization of that compound. At the production level, it's also important for quality control of the drugs, the analysis of the raw materials, the excipients, fillers, any kind of dyes, or any other type of additional material that will go into the formulation. We have to make sure that it's the correct API and that the excipients will not transform the material into something that will be potentially harmful to the patient that is taking them. And the analysis of the commercial product, the final commercial product, is also very important. It's also carried out with, among other techniques, with X-ray powder diffraction, or with, in general, powder diffraction, to detect any kind of changes in the API, monitor the stability of the active pharmaceutical ingredient, any kind of physical or chemical change in the product, aggregation, change in color, for example, which is not very good to have occur in a medication that we are using. Okay. It's also important to determine the illegal or counterfeit formulations, something that is unfortunately common in certain parts of the world. And also problems with patent infringement and things that have to do with litigation processes, because one company claims to own the patent to that particular drug or formulation, and so it could be also a complicated thing, but powder diffraction actually is very important to try to solve those kind of problems. So one of the compounds, one of the APIs that we studied, that we took actually to the synchrotron to try to solve the problem that we have in the structural determination was rasagilin mesylate, which is a compound used in the treatment of Parkinson's disease. It's very, very common, and in Latin America, in particular in Venezuela, this drug is very common. It's commonly used. So it is marketed as the mesylate salt and also as a hemitartrate derivative, but it's mostly formulated as the mesylate salt. We did a search in the Cambridge Structural Database and in the PDF4 organic database at the time. Now it is combining the PDF database is combined into organic and inorganic compounds, but the search at the time indicated that there was one similar compound, but it was not the API that we had on hand, and it was ethane disulfonate of rasagilin, which is the active component of the formulation. And the powder diffraction file contained the calculated pattern from this entry of the CSD. A search in the Google Patents site indicated that there were 2 crystal forms reported for rasagilin mesylate, and there were different experimental powder diffraction patterns that had no particular structural information that we could use to characterize the material. So here is a superposition of the different forms that have been reported. There is a form 1, the first these two materials. This is our experimental pattern, and a form 2, which supposedly has these powder diffraction patterns. But our material, we can see that coincides better with form 2. We attempted structural determination using laboratory data and using as a starting point the rasagilin fragment in the structure reported in the CSD and a mesylate fragment that we found in a structure also reported in the CSD. After a poly fitting of the pattern and using DASH, we obtain a solution, and the refinement of that of the atomic positions without including preferred orientation, it led to an incorrect structure. There was a structure determined kind of satisfactorily based on the chi-square profile here, for example, that is important to follow, but it required using or refining a preferential orientation parameter for the 004 plane, which is which gave this value, which is not very good actually for the preferred orientation parameter refinement. We did the Rietveld refinement anyway, and we obtained these final discrepancy factors, which are not too bad, but the superposition with the structure minimized by DFT calculations gave a value of RMSCD, which is very important to determine if the structure is well determined or not. It is well below 0.35, which has been described by Van der Straat and Neumann as a good value, but this actually showed that it was not the correct structure. The Ripple refinement resulted in these parameters using the DFT minimize results, and but we see that is basically the same. When we, after collecting data at ID22 at the SRF, this is the pattern obtained, and using the DFT model, the energy minimized model with Topaz Academic, we obtain a much better fit for the preferred orientation parameter and a good fit of the different discrepancy factors. And this indicated that in this case, this is just a summary of the parameters. This indicated that we now have the correct structure. We see that the configuration at C1 is R, and that this plane is actually planar. In the model obtained with laboratory data, there was a slight change in the position of one of these atoms, so the configuration of this ring was not the best one. And the examination of the distances and angles with Mogul, one of the programs included in the Cambridge Structural Database set of programs, indicated that all the distances and angles were acceptable, were not out of what it is expected with respect to other structures. The superposition of the experimentally determined structure with the synchrotron data and the energy minimized structure gave a very good value, and we can be confident that this was the correct structure. Other type of materials that we have studied are some anticonvulsants, in particular the derivative of valproic acid. And the commercial form usually contains a mixture of valproic acid and sodium valproate. However, there are some formulations that are used in several countries with other metal derivatives, and we wanted to explore the structures of those compounds that have not been actually well characterized. So we prepared different compounds, different derivatives of calcium, strontium, zinc, and cadmium, and we studied the structure with by single crystal, and also used powder diffraction at ID22 to see the behavior upon cooling the material. All these compounds have some common structural features, and we saw that they displayed a very interesting behavior with temperature. So by single crystal X-ray diffraction using a silver source in the Benemérita Universidad Autónoma de Puebla in Mexico with Dr. Sylvain Bern, we detected that there were one monoclinic structure, the material crystallizes as monoclinic at room temperature with this structure, and at low temperature, as we lower the temperature, there is a transition to a triclinic phase with a smaller cell. In between, we did this study at the SRF at ID22. In between, it's a very, very complicated behavior, but we can kind of pinpoint a temperature where we think that there is a good possibility that this is the transition from one crystalline structure to another. And we are in the process of completing this description, because it's not easy. The behavior with temperature is very complicated, and actually the other the strontium and cadmium materials are even more complicated than this one. This is a setup in a capillary. We use the cryostream for capillaries that can go in this range, and in this link you can see a very nice video of how it works and how the robot works actually. We also study the structure of piperidine hydrochloride, a very complex compound that is also used in the treatment of Parkinson. And one of the problems here is that there were no reports, no previous reports of this structure. We recorded laboratory data, but even though we attempted to solve the structure, it was not possible with laboratory data. There was a lot of preferred orientation in the pattern, but we successfully determined it from synchrotron data using direct methods. And this is one view of the packing arrangement. And the work was done by Daniel Pimiento at UIS in Bucaramanga, Colombia. And we have been collaborating in the study of these particularly different and difficult compounds. Here is a graphic with the pattern obtained with the laboratory source converted to synchrotron scale and the pattern from the synchrotron study. And we can see that there is a much better definition of the diffraction maxima. We have we see better some very small peaks, and so we can better obtain a better description of the structure because we can model in a much better way the preferred orientation. This is another compound that we studied at ESRF, alisapride, and in this case, we also had a report in this case of a monoclinic phase in the CSD, but the experimental pattern was different, and there was a lot of preferred orientation, so it could not be solved with laboratory data, but it was successfully solved with synchrotron data collected at the SRF using direct methods also. And this is just a graph to show the pattern of the monoclinic form and the pattern obtained with the laboratory in the laboratory in Colombia, and the pattern obtained in the synchrotron. And from this this pattern, it was possible to determine the structure with very good indicators of confidence there. So just to finish, I would like to acknowledge the support of FONACYT and the Ministry of Science in Venezuela, the university, and also IVIC, our collaborators there, the laboratory for structural chemistry at Universidad Industrial de Santander in Colombia. We also would like to acknowledge the support of Cambridge Crystallographic Data Center for providing access to the Cambridge Structural Database through the FAIR program. And more recently, we obtained also access through the FACE program of FIS to the ICSD database. Our collaborators at Indiana University, Northwestern University, and Purdue, and the companies that have provided some of the APIs that we use in our studies. Here's a photograph of our collaborators and a picture of Analio and I with Andy and Catherine at ID 22, and some of the people that have worked in some of these particular materials. So thank you very much for your attention, and I'll be glad to answer questions if there is still a little bit of time.
Thank you very much, Graciela, for a very informative session on powder diffraction, and congratulations on the nice results. We have several messages congratulating you. One special saying thank you for the inspiring and well simplified academic talk on the entire subject. Highly appreciated. Muchas gracias. So I'm going to take a couple of questions, and please let me know how would you like to respond. Here we have Felix asking, what is the free access software for analysis crystal phases in XRD? Which other software can be used in place of Expert HiScore?
I'm sorry, can you repeat the question, Carolina?
For sure, it is in the chat. So Felix is asking, what is a free access software for, for analyzing crystal phases in in XRD? And at the same time, which other software can be used in place of Expert High Score?
Okay, well, there are several programs. For example, GSAS2 is open access. It can be used, freely. Dash, also for structural determination, is now free of access. It used to be included in the Cambridge Structural Database set of programs, but now it's free access. And Expo, also from the group in Bari in Italy, is also free access. So all these programs, there is also Fox. These programs can be used free of access. Of course, the programs, the set of programs that are provided with the different diffractometers have a lot of many features that make the process more automatic, easier to carry out. For example, the graphical interface, but there are also some resources available that can be used freely by the scientific community.
Wonderful recommendation. Thank you. And Ahmed is asking, does liquid crystalline material is a quasi-crystals? If not, what is the difference?
Let's see. Liquid crystalline material, if it is a quasi-crystal, no. It is, I mean, it can be studied by diffraction techniques. It can be, well described and represented, but in quasi-crystals, the main thing is that you see a symmetry that is supposed to be forbidden by the conventional crystallography. So quasi-crystals, you usually have symmetries like 5-fold symmetry, 10-fold symmetries that are supposedly forbidden by conventional crystallography, and that requires more than one dimension, more than 3 dimensions to describe it properly, and that's the complication. But no, it's not necessarily the same thing.
Great. We have a growing list of questions. Zakaria is asking how the organic derivatives explored will crystallize prior to X or D measurements. Where and how the organic derivatives explored well crystallized prior to X or D measurements.
Well, yeah, in most of the cases, we try different procedures. We try different solvents and different ways, low temperature, high temperature, hydrothermal conditions, for example, to try to obtain single crystals. But these cases, were not successful in producing single crystals of the quality appropriate for a single crystal study. In one particular, the thing is that they may undergo phase transitions, or they can produce a different polymorph, for example, in the meantime. So we cannot be sure that what we started with is actually what we are studying at the end. So it is a little bit tricky, but yeah, we try many different crystallizing conditions, and we unfortunately were not successful.
Thank you so much, Graciela. We have one last question, I think, because of the time. And Felix is asking, can you explain more on data visualization from XRD and what type of information can be visualized? What is the input data format?
Okay, in general, well, different equipment and different vendors have may have some different formats. but in all the cases, it can all be reduced to what is called the XY format, which is just a list. I can post, for example, in the slides, if you want to see that, an example of that. So you have a list of the peak positions and the intensity, and most vendors of the equipment the software can produce that kind of list that then can be used by different type of programs.
Well, Graciela, they cannot stop thanking to your presentation. Congratulations on a very nice and informative session. If you can see in the chat, there's a couple of other questions that might be answered. They are also requesting for your presentation. So maybe you can follow up with the questions in the chat.